Yeah, the panelists, if you can come up to the front.
Do you have a, it says 5 minutes, it's like 2 minutes, my intro.
Oh, okay. Okay.
2-3 minutes, I mean,
Great.
one slide.
Okay. And it will bring.
So basically I'm saying, thanking the presenters and the patients for being here. And then I describe what the sessions are, the break, and introduce Dr. Nickerson.
Okay. Dr. Nickerson? All of them?
Just Dr. Nickerson.
Okay.
Because he's the first one.
Oh, okay.
Just the first, and then I told them to introduce the next. I'm not actually going to introduce all of them.
Then when we start the discussion questions, then, so I can take the mic and do the movement through here, through the discussion questions, but we'll introduce Kevin Martino, because he won't have been introduced since he's not a speaker. So we'll let him, right before the panel discussion starts.
Okay. Actually, do you want to ask them to introduce themselves before we start presenting?
I don't think it's necessary.
Okay.
You could, in the interest of time, you let them do their introduction.
I'm so busy talking to the reporter. I need to do a bio.
Okay,
Now,
just come.
if you want to start,
just come on. Yes. You know what? Introductions might be. Except he's now taking a bio break. So we'll just do your plan. I like that.
Yeah, I'm going to definitely. Do I say which institution? I always say Dr. Nickerson who's going to talk about this.
They can introduce themselves. You can even say, I'll let them introduce themselves. He'll come up and he'll say.
Okay, if I say, well, please introduce yourself. Oh. Oh, the agenda left here.
Okay, I'll get an agenda.
What happened? Oh, he's keeping it, I guess.
Okay, so then you can call people to order. All right, everyone, we're going to get started now.
Still eating.
Yeah, you can keep eating. In fact, I will be eating as well. It's a short lunch break. And please, another reminder, take bio breaks as you need them. This is a very informal discussion and format here. But I'm going to turn it over to Aslam to give a preface to our afternoon's discussion.
Hello, everyone. Welcome to the afternoon scientific session of the public meeting on patient-focused drug development, which will concentrate on medication adherence and experience with intervention. We want to thank all of the patients, their representatives, and afternoon session presenters for their time and effort. We have a very diverse group of presenters today from various backgrounds, and we're very excited about that. The afternoon scientific session will be made up of 2 sections with 4 presenters in each one. And the first session will focus on causes of late allograft loss and the impact of non-adherence, definitions, terms, and background. And the second session will focus on interventions to mitigate on non-adherence. There will be a panel discussion after each session with a break between the two sessions. and we welcome interactions with the audience during the panel discussion. Our first speaker will be Dr. Peter Nickerson, and he's going to present an overview of late allograft outcomes and risk factors for premature graft loss.
Thank you very much. I want to thank the FDA for a chance to come and to share some of our research that we've been doing. And I want to thank you. I've really enjoyed this morning hearing the patient's perspective, hearing what are some of the key issues that you're dealing with, and in fact, many of the ones that you're dealing with are some of the ones that we're trying to solve. So again, I want to thank you for your input, and we hope for this afternoon. I think the panel is looking forward to ongoing input from you and help guiding us in some of our thinking. So I am going to focus on late allograft outcomes and what we've been learning. In terms of disclosures, I do have some consulting relationships with Novartis and Astellas. I won't be discussing any off-label drugs. You've seen this slide before, but essentially what it's showing us is over the last decade, we've been using a combination of therapies in our immunosuppression. Induction therapy with T-cell depleting therapies has become the dominant approach. About 60% of our patients are receiving this in our centers. Calcineurin inhibitors, tacrolimus, has become the dominant CNI that we're using. In terms of anti-metabolite, mycophenolate, in about 90% of our patients receiving mycophenolate. mTOR inhibitors, rapamycin, for example, had a bit of a presence back in 2003, but it's diminished subsequently. And about 2/3 of programs are trying to go steroid-free, depending upon what type of patient they're dealing with. So this is where we've been for the last decade, and we really have nothing new to offer very much going forward at this point in terms of what combinations we have to offer to patients. So one of the questions is, well, how are patients doing overall? And this is my program in Canada, where we have almost a decade's worth of patients here, 500 patients that we followed. We can see that half are from deceased donor transplants and half are from living donor transplants. None of these patients had donor-specific antibodies pre-transplant. We'd ruled that out with a cross-match. And we were seeing acute rejection rates in the first year of about 11%. And I think this is pretty typical of most programs nowadays. And when we do death-centered graft survival at 1 year, 99% of our kidneys are surviving. And at 5 years, 96% of our kidneys are still functioning. So again, I think this is fairly typical data that are coming from most centers over the last decade. But what we are seeing is that patients are losing their kidneys or developing dysfunction in their kidneys beyond those first few years. And this is a long-term follow-up in this cohort where we've looked out as far as 15 years post-transplant. And we actually see 3/4 of our patients, 76%, are actually having very stable function. They're doing well on their immunosuppression. But the other 24% have been developing some problems. And I'm going to deal with a few of these causes. 11% are other causes, and those other causes are recurrent kidney disease. So one of the common things we see is recurrent IgA nephropathy in the kidney. We see some isolated T-cell mediated rejections late in about 2% of our patients. IFTA is another way of saying scarring or fibrosis in the kidney. We see that in about 1.6%. Infection in the kidney caused by a virus called BK virus, leading to kidney damage, 0.4% of our patients. And then there's a smattering of others. But the largest thing we've been seeing is that about 13% of our patients are developing donor-specific antibodies against the mismatched antigens in the donor kidney at some point post-transplant. 4% of the time, this is showing up with an acute rejection. So this is graft dysfunction, and we detect an antibody in the blood against the donor. And 9% of the time, this is occurring subclinically. The kidney's functioning fine, but we find that there's an antibody in the blood when we screen the blood. And these patients subsequently go on to develop dysfunction at some point down the road. We do see death with function. We have 10% of our cohort in this study that had died with a functioning graft, and most of those patients, 9% of them, had died with a stable functioning graft. Now we expect, and our goal is death with function. We want you to live the rest of your life with a functioning graft. What we don't want is premature death with a functioning graft, and that is some of the issues that we'll talk a little bit about later. In terms of the incidence of donor-specific antibodies post-transplant, in our program, what we see is that about 2% of our patients have developed a donor-specific antibody by a year post-transplant, and by 12 years post-transplant, about a quarter of the patients have developed this antibody. Now, there's other groups that have shown higher rates, 20% in the first year and 5% per year thereafter. But again, there is some discrepancies here, I think, in how people are defining these antibodies using the diagnostic tools that we have. And I'm not going to go into any deeper detail in that, just to say that the true incidence is probably somewhere between these ranges. In terms of what are the risk factors for developing an antibody after the transplant, it turns out how mismatched you are is one of the driving factors. When we first did transplants, and this is one of the first transplants in the US done in 1955 at the Brigham and Women's Hospital, where they had identical twin transplants, we didn't need any drugs. These were truly identical genetically at all genes, and so you don't need immunosuppression when you're HLA identical. What we've learned from the UNOS database, even into 2004, was that if you are mismatched at the class 2 region, HLA-DR, you're at a higher risk of having graft failure late post-transplant. So we know that the better you're matched with your donor, the better you'll do. The other thing that we've been learning about these antibodies when they're forming is that they're largely against what are called HLA class 2 molecules, the DR or DQ molecule. And HLA class 1 mismatching doesn't seem to drive de novo DSA formation quite so much. The other thing we found out was that T-cell mediated rejection, so a cellular rejection early post-transplant, if you have this kind of a rejection, it seems to put you at higher risk for subsequently developing an antibody somewhere down the line. And that may just be relating to the fact that you're allowing the immune system to turn on, and at some point it turns on enough that it allows an antibody to be generated. And many groups around the world now have shown this association. So we were showing this back in 2012 in Canada. Groups in Europe, Japan, and in the US have also found this relationship that early cellular inflammation in the graft can lead to the subsequent development of these antibodies. One of the things that we've also been trying to do, and I think it's related to a lot of what we've been hearing, is that we want to try and minimize our drug immunosuppression. We want to try and reduce it so that we can reduce our side effects. So this was a study that was funded by the NIH. It was a multi-center study in US and Canada where we had patients enrolled who had received living donor transplants. We made sure that absolutely at the time of transplant they had no evidence of any memory in their immune system towards the donor. So this is truly going into what we call a naive immune system. We gave them what is considered standard of care in terms of immunosuppression. They had thymo as an induction therapy and then received tacrolimus, Cellcept, and prednisone. And for the first 6 months, these patients had absolutely no acute rejection. We did a biopsy at 6 months to look in the graft to make sure that the kidney was actually doing fine, and the histology here was completely normal. And we tested these patients for antibodies to make sure that their immune system hadn't started developing an antibody against the mismatched antigens. And then we randomized the patients into coming off of their Cellcept, their tacrolimus, over about a 3-month period. So we did a slow taper, watching them very closely to see whether they were going to develop rejection or not. And what we found was that as we started withdrawing the tacrolimus, we started having some acute cellular rejections, and we started developing some donor-specific antibodies. And this was occurring frequently enough that the DSMB, the Drug Safety Monitoring Board of this clinical trial, halted the trial. We were supposed to enroll well over 300 patients into this trial, and they halted it after 21 patients, saying, this is too frequent an event. You cannot continue this trial. We don't feel it's safe to do so. And what that taught us was that just because you're doing well on the immunosuppression that you're on doesn't mean that reducing the immunosuppression is necessarily a safe thing to do. And that's a major message that the community had to learn. And I think because we see patients in our clinic and we say, you're doing quite well. Why don't we try lowering your immunosuppression a little bit? And I think that's a slippery slope that we can get into, thinking that you're doing well, and all of a sudden we get below that level. and we see the immune system's capable of mounting a response and getting activated. And that's not what we want to have happen. We look at non-adherence, and we found that this was a major risk factor for developing antibodies. And so in patients who had reported to us that they'd been missing drugs, that their rate of developing antibodies actually was about 72% at 12 years, compared to only 19% in patients who were taking their medications as they were being prescribed. So this is a major risk factor that we had identified. And the other question is, well, when does non-adherence occur? And this was an interesting study out of Minnesota, where they had 195 patients start using what are called medication event monitoring systems. So in the pill cap bottle, there's a little microchip, and it time and date stamps when you open the bottle and when you presumably then have taken your medication. And this was an informed consent study. They told patients, we're doing this to see how well you're taking your medication. And what turned out to be true was that 22% of the patients were starting to miss 7% or more of their drug doses by 2 months post-transplant. So non-adherence and missing drug medication is not something that happens years after the transplant. It actually starts early post-transplant. And I think this just tells us a couple of things. One, that missing your drug is enmeshed in a very complex world. You're living your life, you're trying to remember to take your medication. We heard about that earlier. And life, I think, gets in the way, and it's one of the harder things, and we're going to talk a lot about how we can help try and avoid this. But what this translated into for these patients was that they started having late rejections, so a year to 2 years later after the transplant, they were having more acute rejections in their transplant. And by 3 to 5 years post-transplant, they were starting to lose their grafts more frequently than those patients that were adhering with their medication regime. When we put this all together in a multivariate analysis where we're trying to look at what are the independent predictors of risk for forming these antibodies against the graft post-transplant, what we found for antibodies against HLA-DR was that it was non-adherence, how mismatched you were to your donor, and if you had these early cellular rejections preceding the development of antibody. For HLA-DQ antibodies, again, it was non-adherence, how much you were mismatched to your donor for HLA, and the recipient age. And what the recipient age here was that the younger the age, the more likely you were to form these antibodies. And this was independent of non-adherence, because a lot of times younger individuals are always being said, well, they're more likely to miss their drugs. That's got nothing to do with their adherence or non-adherence. It's got to do with their younger age. At a younger age, you probably have a more robust immune system, and therefore you're more likely to respond to mismatched antigens. When we looked at what was going on when these antibodies were formed, and I'm not going to drag you through a lot of this in detail because it's going to get into a lot of what are called histologic grading systems, but suffice it to say that 76% or 3/4 of our patients that were forming these antibodies in their blood actually had histologic evidence of rejection in their grafts. So there was a good correlation between the detection of the antibody and evidence of antibody-mediated rejection inside the graft. There was also evidence in these patients of having T-cell mediated rejection concurrently. Only 18% had no evidence of rejection. So just because you have an antibody doesn't always mean that you have rejection, but certainly the majority did. And we started to see that there was some scarring also in these kidneys by the time that we were doing these biopsies. And we think that that was probably related to the immune inflammation going on inside these grafts. Once they form these antibodies, if we look at time 0 as the time we first detect these antibodies, if you started out having what's called the subclinical rejection, where the antibody was there but your graft was functioning fine, it took about 8 years for half the patients to lose their grafts. And if you had a clinical onset of a rejection at the time that antibody first showed up, it took about 3.5 years before you would lose your graft. So this wasn't a sudden process of losing your graft, but there was a slow progression to graft loss once these antibodies were forming in a fair number of these patients. When we looked at what's the strongest predictor of fibrosis and scarring inside the filters of the kidney, we saw that it was the formation of these antibodies. And when we looked at what predicted whether or not you form scarring in the interstitium compartment, in the tubules of the kidney, we found that that was related to early cellular rejection and again to non-adherence. So again, thinking that non-adherence was leading to inflammation inside the graft. So this brings me to the summary slide, which is when we do a transplant, I'm sorry, I don't have a, I guess I do have a pointer. When we do a transplant, there's a lot of factors that are going into how long that graft is going to last, or whether or not our patient is ultimately going to pass away with either hopefully at a natural lifetime, but we know that premature graft loss can lead to premature death. Scarring inside the graft largely relates to a number of different things, but one of them is certainly rejection, cellular rejection or antibody-mediated rejection, and the antibody-mediated rejection is preceded by the formation of these antibodies. And the degree of mismatching between the donor and the recipient is really what's driving the immune system to go forward. We try to counteract that by giving immunosuppression, but the immunosuppression itself has some toxicity that we've been talking about, and it has side effects that put you at increased risk for metabolic problems like diabetes, infection, or tumor. And we can have recurrent disease or infection in the graft, and we have also the comorbidities, the illnesses that everybody has prior to developing their end organ failure that can be contributing to the outcomes. We know that non-adherence acts as our inadvertent minimization of immunosuppression can lead to acceleration of the immune response towards the graft. And so this is why we've been focusing a lot on non-adherence and minimization, avoiding minimization inappropriately. What are the keys to the future? Well, I think one of the keys is for us to think about how can we match better when we have an organ available. How can we dial in tissue matching beyond what we're currently doing now to try and, if we were to do that, it would decrease the drive for this rejection response, which would mean that we could use less drug. That's one strategy going forward. The other key is how do we actually understand better what is non-adherence, what leads to non-adherence, and how do we help the patient avoid non-adherence? And then the third thing is we need new drugs that have better immunosuppressive profiles with less drug toxicities and side effects, as we've been discussing already today. And with that, I'm going to stop. Thank you very much. Our next speaker is Dr. Rita Alloway, who's a research professor of medicine and director of transplant clinical research at the University of Cincinnati. Rita.
Thank you. First, I would like to take the opportunity to truly thank members of the FDA and the patient community on behalf of the transplant professionals for the spirit in which you have come today and allowing us to better recognize the issues that are very, very important to you. And it, even though we know it, it helps to hear it yet again. I am going to lead in the next discussion in terms of talking about the aspects of non-adherence, focusing on definitions and identification, and also looking at the detection and risk factors. In terms of financial disclosures within the last 12 months, my institution has received several clinical research grants from a variety of sponsors of a variety of studies in transplantation. I'm on the advisory board for Genzyme Sanofi and on the speaker bureau for Aloxis and Sanofi. This presentation does not include discussion of off-label or investigational use of any drugs. So the objectives I want to focus on today is to differentiate between medication non-adherence and compliance. I want to identify risk factors for non-adherence in solid organ transplant recipients, and I want to describe the measures to quantitate medication non-adherence. As we all know, medication non-adherence is an age-old problem. Hippocrates was actually credited with a statement in 500 BC, keep watch also on the fault of patients, which makes them lie about taking things prescribed. And as not to offend anybody's religious beliefs, I might want to offer non-compliance may have actually started with Adam and Eve when she took the bite of the apple, but we won't go there. C. Everett Koop in 1985 went a little step forward and basically trying to explain the variability that we see in drug responses. Why does the drugs not work the same in all patients? And basically he said drugs don't work if people don't take them. Now I think that Typically, non-adherence, especially when we describe this to you as patients, it sounds like a very negative connotation, but I want us to let down those barriers and encourage you to believe that we understand why you are non-adherent sometimes. However, we are still here to try to help you and hopefully develop things in the future that will improve this. And I think what's evident in transplantation is that we can no longer accept the status quo of any non-adherence that is occurring. And to follow up with one of Dr. Nickerson's quotes, as he has said many times before, is our first shot is our best shot for transplant success. I know many of you here today have talked about having the beautiful medical miracle of multiple transplants, but really our first shot is our best shot for biologic reasons, as he discussed, with the formation of antibodies, to the additional comorbidities that come as we begin to add on the stronger immunosuppressants that you take with each transplant. Unfortunately, despite millions of dollars in investments, we don't have a magic drug or procedure to render adherence irrelevant that's currently on the horizon. So what are we going to do for the next generation of transplant recipients? And hopefully adherence does not continue to be neglected in the therapeutic process, and to such an extent that federal mandates may be necessary to properly resource adherence incentives. We want to provide the resources to facilitate good medication adherence. Now, there are a variety of terms used to describe taking your medications appropriately. Two of the most common terms of medication adherence and medication compliance. Medication adherence is the extent to which a patient takes medications as prescribed by the healthcare provider, while compliance is described as more of a passive act of the patient following the provider's orders. In a 2012 publication in the British Journal of Clinical Pharmacology, they have a really good summary article about the taxonomy and definitions of medication non-adherence, and why they fundamentally believe that these two terms are interchangeable. They feel like compliance focuses too much of a passive act of the patient basically following specific directions that the provider may have given to them, while medication adherence connotates a more collaborative approach with the patients and the healthcare providers. And hence the words medication adherence have now become kind of our go-to terms, at least as present, to discuss this concept. And medication adherence is a behavioral process that is influenced by many, many factors, many of which we've heard today. It assumes that the patient has knowledge, motivation, skills, and resources to follow the healthcare provider's prescription. Medication non-adherence can fall in two different categories. There can be intentional medication non-adherence, which is basically an active process by which the patient chooses to deviate from a treatment regimen, or unintentional medication non-adherence, which is more of a passive process where patients may be careless or forget about adhering to their treatment regimen. Now, the World Health Organization defined 5 dimensions of adherence, and these dimensions included health system or health care team factors, social or economic factors, condition or comorbid disease factors, therapeutic or medication factors, and also patient-related factors. Our next speaker, Mary Amanda Dew, has basically published in 2007 a meta-analysis which basically reviewed the literature that was available at that time to look at the specific factors that had been reported in transplant recipients and categorize them in these dimensions. In terms of the socioeconomic factors, younger patients, male gender, non-Caucasian, non-U.S. residents, poor social support, poor transportation, and literacy were common factors that were limitations in terms of the social and economic factors. In terms of the therapy or the immunosuppressive factors, complex medication regimens, higher medication toxicity, as we've clearly heard, lack of medication education systems, and lack of pillbox or reminder systems were therapeutic-related factors that are common in transplant recipients. Patient-related factors were that a history of non-adherence tended to predict the future non-adherence with the immunosuppressive regimens. Adolescents, psychological disorders, as many have discussed today, related to depression, cognitive impairment, substance abuse, and negative beliefs of the medications were contributory factors to non-adherence. Condition-related are basically high symptoms of distress. And I think, again, it's a common theme that we've heard today. The new disease that you have of immunosuppression is a stressful disease, and it's something that you must manage. Also, we heard loud and clear, development of new onset diabetes, or basically diabetes that occurs in you after you're transplanted that you did not have before transplant. And also your increased time post-transplant. Healthcare system factors such as medication cost, poor access to medication, poor aftercare planning, poor physician-patient relationship, and poor physician communication, again related to the healthcare system and healthcare team factors that led to non-adherence. So anytime you do a risk assessment analysis, you want to look at all these figures and factors and attempt to differentiate which of these factors are modifiable and which ones are not. Any of these factors in which are modifiable, we want to try to develop a system approach to address these. However, as we're focused today on patient drug development, you have to understand that for us to really have an impact on drug development from an adherence perspective, we've got to be able to measure at least pharmacoadherence, your adherence to your medication, in a proper way. When you attempt to measure pharmacoadherence, you have objective measures, which may be direct and provide evidence that medication has actually been taken and consumed. Examples of that are actually direct observation, and we have one drug available in transplantation where we do that routinely. We have indirect measures of adherence, such as providing evidence suggesting that medication is being consumed or taken, such as pill counts, tacrolimus drug levels, pharmacy refill records, medication possession ratio. And then lastly, subjective measures, which provide testimony that the medication has or has not been taken, often by self-reports or assessment of others. So in terms of direct observations, this is basically has the advantage of being objective, highly specific, and non-invasive. However, there are disadvantages. Feasibility issues, they're very labor intensive, they're not practical, they may be expensive, and they're not actually an option for all transplant recipients. Drug concentration monitoring, which we are very fortunate in transplantation, really since the development of calcineurin inhibitors, to have drug concentration monitoring available to us. The advantages of this are it's objective, it's part of our standard of care, and direct assessments of whether the patient has taken the medication, at least during that short period of time, is available for us to review. Disadvantages are that this is really just a snapshot of the behavior of what's going on as we look at these levels. And unfortunately, it can be affected by a variety of factors other than pharmacoadherence, such as the way you metabolize or get rid of a drug-drug interaction, a drug-food interaction, and poor absorption. And I think that historically, before the broader understanding about all of the factors that impact drug level monitoring, we tended to maybe, quote unquote, assign non-adherence to a lot of the variability that we saw, which may be attributable to other factors. And even though it's a very, very good marker and we can understand if the drug is in your system, there are a lot of other factors that we have to consider. It's also costly and it's an invasive test. However, some more novel approaches have recently been introduced to look at the tacrolimus level variability instead of just a single snapshot. And without going into the complexities of the study, basically patients that had a higher tacrolimus level variability over time, as measured here by standard deviations, basically were associated with negative risk factors or negative outcomes such as late acute rejection, transplant glomerulopathy, which is a negative biopsy finding that's previously been described, or total graft loss. And even when we excluded death with function, variability that was associated in the tacrolimus levels that we monitored still predicted these negative long-term outcomes. Another form of monitoring is actually electronic monitoring. It does have advantages of being objective. It can indicate the date and time a bottle or pill box has been opened, providing you a real-time tracker. With this specific information, it also can detect poor pharmacoadherence with a dosing schedule. So for example, you may do very well with your morning dose, but very poorly with your evening dose. And it allows us to get an idea of what is working and what isn't working in regards to your medication regimen. The disadvantages are cost, among others here, and basically it assumes that medications that are removed from the bottle are actually taken. Strategies to impact non-adherence have been incorporated into some recent publications, again focusing on the electronic medication monitor, the MEM system that Dr. Nickerson previously described. So for sake of time, I'm going to skip over this. One of the other measures that we have of medication adherence are refill or pharmacy records. When we actually take the records from the pharmacy of the number that was dispensed, the date and time that was dispensed, and the dose that you may be taking to provide objective standardized data to identify patients who may not be adherent. The disadvantages of these systems are possible misinterpretation of the use when making dose changes, which are very common in transplantation, as you know, and that's one of the real limits, especially with tacrolimus, of us effectively using these records. It assumes that prescriptions are filled or actually taken. It assumes that all sources of medication are captured and that there is increased complexity when there's using multiple pharmacy records. This data has actually been translated to a medication possession ratio or proportion of days covered, and basically use these formulas to estimate the adherence to chronic medication. Now, what is very frequently brought up in transplantation is why we tell you you must be 100% compliant all the time. We really don't know what the optimal medication possession ratio is for any of our known immunosuppressants to date. And then there are self-reports, which I'm sure many of you have been asked to fill these out. And these can be, unfortunately, quick and inexpensive, but unreliable. But if you think that the self-reports are unreliable, you should even see how worse reliable the clinician reports are that we provide. And we may think it's simple, quick, and inexpensive, but it is highly inaccurate. So in summary, an article, a chapter published recently in Clinical Transplants by Dr. Tiffany Kaiser summarizes these methods to monitor immunosuppression and focuses on many other aspects of non-adherence. So in terms of quantitative non-adherence, there are many measures of pharmacoadherence that are applicable to transplantation. However, there's no single perfect measure of pharmacoadherence. Multiple measures of pharmacoadherence are optimal to provide an accurate adherence assessment, and this is going to be necessary for us to introduce it in the future of drug development for transplantation. Okay? Thank you very much. Our next speaker will be presenting the prevalence of non-adherence after organ transplant, and it's Dr. Mary Amanda Dew. She is professor of psychiatry, psychology, epidemiology, biostats, and clinical translational science. She's the director of clinical epidemiology program at the Western Psychiatric Institute and Clinic at the University of Pittsburgh School of Medicine. Dr. Dew?
Thank you, Rita. Well, I'm really humbled after hearing all of the comments of everyone this morning. So I hope that what I will tell you will be informative, and I think that actually some of the evidence really supports all of the things that you've said, and you might be pleased to know that some of it has appeared in the scientific literature so that other healthcare professionals may have been forced to read it. So on that note, I'll begin. But only if I can make this move. Okay, so when we look at prevalence, we're talking about counting things, and counting can be pretty boring. So why do we have to count how many people are non-adherent? We need to know exactly how common non-adherence is in order to estimate, in order to look at the scope of this problem, to estimate how many people are likely to have trouble with various elements of the regimen. And we need to know how common the exact prevalence in order to design and test interventions that are targeted to the right people and are cost effective. So adherence is really a breadbasket of different activities. It's not just taking medications after transplant. You all have spoken about the various other things that are required after transplant, and there are many things, and that's part of what makes this a complex problem. There are two ways to study adherence. You can look at it from a quantitative perspective where you ask patients to report on their adherence. You can use biologic measures, all the approaches that Rita was just describing. Or you can use qualitative measurements where you rely on patient descriptions of how they manage their problems and what kinds of problems they experience. And in fact, there are many studies within both of these categories, and there have been now several definitive systematic reviews that have tried to summarize all of this evidence so that we can have an overall picture of the nature of this problem. So I'm going to talk about those reviews today because there are too many individual studies to make sense of them on their own. So our group in Pittsburgh did 3 systematic reviews. We did 3 meta-analyses, and we focused on post-transplant adherence to all areas of the regimen. We looked across all types of solid organ transplantation, and so we examined adult general transplant samples. We looked at a subpopulation composed of people who are transplanted after histories of substance abuse, which is quite common, for example, in liver transplantation, and we looked at pediatric studies as well. So this shows for the adult and pediatric studies, there were 147 studies of adults, and that's on the left, and you can see that most of them involve kidney recipients, which makes sense because that's the most common type of transplant. And then heart and liver recipient populations were the next most common. In the studies of kids, kidney recipients also were the most common, but liver was the second most common. And these studies came from all over the world, but primarily from North America and Europe. And then the studies that focused on relapse to substance use were mostly in the liver population. So this slide shows then the distribution of areas of non-adherence that were considered in these studies. And the length of the bar means more studies were done. So the issue of taking medications has been most commonly looked at, and you can see that the bar is about half and half, green and blue. The pediatric and adult studies were about equally as common. But for the other areas of the regimen that you can see down on the left-hand side of the slide, there's been much more study of those problems among adults than among pediatric recipients, even though many of those areas are equally important for the pediatric groups too. I'm going to walk through this slide because this shows the key results from these meta-analyses. So you can see on the left the different areas of the regimen, and then these numbers are the estimates across all of the different studies combined. So if you look, for example, at this first group, if you saw 100 people during a 1-year period after transplant, it might be the first year, it could be the 10th year, whatever, you would expect to see 3.4% of them go back to using tobacco during that 1-year period. With alcohol, it's 3.6% who would go back to using alcohol at levels higher than were prescribed. So these problems, substance uses of different types and attending clinic appointments, are relatively low. They're on the left-hand side of the slide. So not that many patients have trouble in those areas. These ones down at the bottom are the lifestyle kinds of activities. Patients are told they should exercise, follow certain diets, have blood work and tests, and so on. These are more common, so up to 25% of people seen within a 12-month period would have trouble with their diet, for example. And then unfortunately, taking immunosuppressant medications was up in among, it's like the lifestyle things, it's a little bit more common than we would hope. So about 23 out of every 100 people seen during a year would have trouble taking their medications that would rise to a level where they would be considered non-adherent. Then adding on the pediatric information, what I just described to you was for adults. The pediatric rates then are shown in green, and you can see that generally the rates are lower. It's important to note that this includes all age pediatric samples. So, you know that parents have a big role for younger kids. non-adherence to medications goes up a lot among the adolescent group. So that's one important distinction here. But overall, the rates tend to be lower in pediatric groups. So then what are the risk factors? We tried to look at them across all of the studies in our analyses, and actually we could not look at very many because very few studies have looked at risk factors. They often will report on rates of non-adherence, but then not go very far beyond that. But in our meta-analysis in adults, we found the ones that are shown on the left-hand side of the slide. For example, for immunosuppressants, non-white ethnicity, having poorer social support and poorer perceived health were linked to having non-adherence problems. But the important thing is that those linkages were relatively weak. The size of these effects were small. It's not like they were the major factors that anybody would point to and say, oh, I've solved the problem, just focus on this factor. The only one that was really important was that people with pre-transplant histories of substance abuse are more likely to have problems with that post-transplant. But you would expect that because that's the nature of addiction. So that just goes hand in hand with that kind of issue. For kids, we couldn't look at non-adherence by specific areas because there were fewer studies. But we did find that in general, factors like the stress of the parents, the behavioral functioning of the child, and the distress of the child were relatively important. But again, the sizes of these effects were relatively modest, which means that we can't point to a single factor and say that's the cause of the problem for everybody. Now, I mentioned initially there's also been qualitative information. And in these qualitative studies, the investigators focus on trying to capture what the recipients tell us in their own words about the medical regimen as well as other areas of transplantation. And if you look up these three papers, at the bottom of the slide, the dates should be 2009, 11, and 16. So in case you're interested in finding those papers. They focus primarily on kidney recipients, and they have looked at adult recipients as well as adolescent recipients. So in these qualitative systematic reviews, they found that looking across this whole body of qualitative literature, there are quite a few studies that have tried to look at this and get the patient's own words. The first important theme was empowerment, that patients want to gain a sense of control over the regimen. And many people here today have mentioned that, the importance of being able to manage, being able to organize, how a person can organize, who can help. So these are two illustrative quotes. I discovered the possibility of maintaining control, even if you have to ask for help. So they didn't have to do it all by themselves, but they could manage. I'm good at planning ahead. I got this chart, this box I refill once every week. A second theme was fear of the consequences. And you all have also discussed that quite a bit. Fear of graft loss, fear of adverse effects, and defining what is an acceptable risk that they can live with. So one person said, I do think we walk on a knife edge all the time and you can just fall off of it and lose the transplant. And this person was talking about the difficulty of trying to have a life and manage all of these things and the fear that it wasn't going to work. Then another person said, to find out that I had cancer due to the medications would probably be more devastating to me than having kidney failure. So for that person, defining an acceptable risk was that they had an overriding concern about cancer, and that concern was greater than their concern about the loss of that graft. A third theme was managing regimen demands, forgetfulness, side effects, lifestyle disruptions. So people like you have said things like the hardest thing is if you're someplace new or doing something new and remembering to take your medications. Another person said, I really had to push for a medicine change because the doctors didn't think that hair loss was kind of a relevant thing to worry about. So these lifestyle disruptions can be things where the patients do have to push their providers to convince them that this is important to them. This is part of balancing what it means to have a transplant and go on with their lives. A fourth theme had to do with over-medicalizing life, fatigue at being a patient, or self-management burnout. One person who was very demoralized, said, "You can't call it living a life. I'm still living like a patient. I can't do the stuff I wanted to. I'm just dead." So that person really was burned out at that time. Someone else said, "I was really doing well. I started thinking, I don't need all those pills. I just stopped taking them little by little. I was tired of them. They made me feel like a sick person. And then, of course, I went into rejection." So the person realized that that wasn't the best choice, but they did it because of their other concerns and feelings of distress. Fifth theme was social accountability and motivation, and I think many people have expressed that theme here today as well. This kidney was given to me by my wife. I have an obligation to take good care of that kidney. You can't forget your meds. I'd be afraid to face my doctor if I did that. They don't say much, but it's the way they look at you. You know they're disappointed in you. And all of us have been a patient, and I've said that to my husband. My doctor's going to be disappointed. So the authors of those systematic reviews felt that those themes really were then reflected in 5 different kinds of behaviors that were observed and that patients talked about when they did these interviews. First, on the left, was not taking medications, just refusing. Sometimes they refused, sometimes it was inadvertent, it was forgetfulness, sometimes it was side effects, but that fell into the non-adherence level of behaviors. On the other side is total adherence, where people are extremely vigilant, and I think many of you who are here today are motivated by your extreme vigilance and by the fact that you've been able to master this and that you're hopeful that you can help other people to rise above some of these problems and maybe address some of the problems so they can master the regimen as well. Then in the middle is maybe where many patients lie. They want to be completely adherent, but they may be changing doses to minimize side effects. They may forget a dose, or they may vary the timing of doses inadvertently due to other lifestyle factors. So in general then, what can we conclude from all of these reviews of the quantitative and qualitative literature? Non-adherence occurs relatively often. The rates are higher in adults. They're lower in kids, but if you want to look at the areas of greatest problem, it's clinic appointment attendance. Non-adherence is modestly associated with psychosocial risk factors, the things that I showed you on that one slide. But a limited range of such factors have been considered, and the effects are not large. Patients most commonly voice the need to take control of the regimen but not let it control them, concerns about adverse effects, and motivations for following the regimen, and that is what you have said here today. So that it appears in the literature too, and I think that's very important that it's gotten into the literature. And finally, if those of us who are professionals and work in this area would take time, I think, to attend this kind of meeting, we could listen and then maybe generate some new ideas for better ways to address this problem. So thank you. So I'd like to introduce Robert Edinger, who is a distinguished professor emeritus, the Department of Pediatrics, Division of Nephrology, the Mattel Children's Hospital at the David Geffen School of Medicine at UCLA.
Thank you very much, Mary Amanda, and thank you both to the FDA for inviting me. I'm humbled to be here in the presence of so many, real luminaries in the area. I'm just a pediatric nephrologist, but I most of all want to honor all of you who who have really shared with us today a lot of what it really means in transplantation, and something that we physicians really sometimes unfortunately lose a bit of sight of as to the challenges that you're going through as well as the successes that you've had. I want you to focus today. I'm going to try to go relatively quickly, so I'm going to ask that you focus when you do on those things that are highlighted in red, because those are the important things that I want you to see about with regard to how adolescence and pediatrics differs from what you've heard so far. Many of you do not know how to define adolescence, and allow me to do it as a pediatric nephrologist. Adolescence is the age at which dad becomes a two-syllable word, where they go, dad. So just thought you just might want to know that. Adolescents have the best one-year graft outcome. This is data from the Naprotix database. But patients who are transplanted age 11 to 17 years of age have the worst 5-year graft survival. And most have attributed this in large part to the issues of medication non-adherence in adolescents. After the first post-transplant year, adolescents have the highest graft failure of any age group. Now, this is a seminal study by Beth Foster in 2011. And what this is showing, and let me address this specifically to this area right here, because this is the relative risk of losing a kidney allograft at a specific given age, not when you were transplanted, but at a specific given age. And as you can see on the slide, things start to get bad at age 10, 11, 12 years of age, and then continue to go up to where they hit the maximal graft loss in the late teens and early 20s. This is true whether you count or don't count the first year of graft outcome. The point of this is that it's the age at which a patient is that really seems to register with graft failure. So our patients who are transplanted adolescents, they will tend to do badly, but patients who are transplanted as young children, we expect will transit through adolescence, and those patients are equally at risk after a period of relative quiescence. The graft failure that we see is most often due, as Peter said, to antibody-mediated rejection, likely secondary to medication non-adherence. And this is important. The donor-specific antibodies that Peter discussed that are generated during adolescent graft failure due to non-compliance lead to ultimate prolonged waiting times for a second graft and subsequent poorer retransplant outcome. So the first kidney is the best kidney, and we really need to pay a lot of attention to patients who are in their adolescence, because this is going to color their experience for the rest of their lives if in fact they do develop donor-specific antibody. The prevalence of medication non-adherence in pediatric transplant recipients is shown here, and you can see that for liver and heart it's in the 30 to 32% range. Liver and kidney, 30 to 32% range. Heart is a bit less. But when you look at the percent of patients, and this is data that I'm showing you here for renal outcome, less than 10 years of age, it's 22%, greater than 10 years of age, 43.2%, showing you how much more adolescents are at risk. These are the selected factors that are associated with medication non-adherence in pediatric, but shown in red are the adolescent transplant recipients. And specifically, there are issues of low self-esteem. Patients don't like to carry the medications with them and look different. Adolescents tend to have a busy lifestyle and are sometimes disorganized in terms of being able and forgetful and being able to remember to take their medications. We've heard a lot about the psychological issues this morning that may afflict patients, and this is certainly true in adolescents. We'll talk a bit more about those psychological issues of depression, PTSD, anger, and denial a bit later on, but poor coping mechanisms can very well be seen in adolescents who are not particularly good at coping anyway. And then finally, there are issues of social skills, poor social skills, deficient social support, and reluctance to admit to friends or peers that in fact they have a transplant. This is a MRI of busted. These are the unique psychosocial and developmental aspects of adolescence. You can see this is the adolescent brain, the love lobe, the rebellion center, all of you see this. I want to direct your attention to this area, right down here, right over here, memory for chores, homework, and we have actually mapped non-adherence and meds. We have scientifically mapped this. Not. Thank you for that. Can I get a... Okay. So what are some unique psychosocial and developmental aspects of adolescence that impact non-adherence? Well, three prominent characteristics of adolescent behavior are risk-taking, just think back to your own adolescence, increased sensation seeking, and the move away from parents to greater peer affiliation. As I've shown in red, that is an important point that we need to be addressing. With regard to patients' cognitive and emotional neuronal networks, adolescents develop differently. Their emotional system circuitry develops earlier. The prefrontal lobe circuitry, which is the executive functions, the able to abstract, that develops later. And the prefrontal lobe circuitry is necessary for executive functioning, which means abstraction, long-term planning, attention, response inhibition. Adolescents don't do these things well because they haven't developed the appropriate circuitry yet. Finally, there's the issue with the adolescents, the need for separation and individuation. And adolescents will tend to experiment to see which values of patients they will adopt. Adolescents will question authority. And the question always is, what happens if meds are missed? If there are no immediate consequences, as we heard this morning, well, I can get away with it now, maybe I can get away with it, a bit later. And thus, the medical team, the docs who say, don't miss your medicines, we lose credibility when adolescents are non-adherent without consequences. Shown here is a scale called the Barriers to Adolescent Medication Adherence, which is actually quite a useful tool. I've listed 4 barrier types, but in red, I think the important barriers for adolescents that I'm showing in this slide are, number one, that the adolescent-reported barriers, almost 30% reported forgetfulness, like not paying attention, I'm completely out. Also, poor planning or poor scheduling. And notice that parents notice this even more than the adolescents. 68% of parents reported that their adolescent transplant recipients had problems with planning, and 58% of adolescents reported that. Now, with regard to the barriers of adherence, in the interest of time, I'm going to direct you to the area in red. Barriers reflecting disorganization or not planning ahead, and the desire to avoid others observing you taking your medications is a major issue. The kids don't want to be found out. This is strongly associated with medication non-adherence and with emotional distress, such as I mentioned, anxiety, depression, etc. These, in turn, these emotional distressors are correlated with medication non-adherence. So it's a cycle. And so the more that these barriers are showing up and we can assess them, perhaps the better we can address them prospectively to keep kids from going on. The barriers tend to remain stable over time, but poor adherence to medication is associated with a series of these barriers that can be measured during the time of transplant, and it's one of the ways now we're using in the clinic to assess how much we have to worry about the kids. What about measuring adherence in adolescents? Directly observed therapy can become cumbersome and contentious between parents and adolescents. The kids don't like taking the medicines in front of their parents. So directly observed therapy is an issue. Success with indirect measures, such as drug levels, is something I'm going to show you just a little bit to how we can perhaps pick up non-adherence early. Electronic measurement systems seem like a good idea, but they can be limited if patients don't want to bring a separate electronic container or smart pill box with them. And finally, self-report instruments are particularly limited in adolescents. This is the results of a liver transplant study by Al Shemish and all called the MALT study. And basically, the point here is that if children have a large variability in their tacrolimus levels, they're more likely to have liver transplant rejection. So these are a robust predictor of late allograft rejection, and following this in the clinic can inform our ability to hopefully head off rejection and address the non-adherence. Finally, this is data that we have just published that's still not out in print, but is on the internet. And this slide shows patients with high levels of high coefficients of variation for combinations of both tacrolimus and sirolimus, shown in the red circle. Those patients that are in this area are patients that have very high variability in both their tacrolimus and their sirolimus levels. Previously, we have also published that high tacrolimus levels in and of themselves predict rejection. But importantly here, what this data shows is that in patients that are on this dual therapy, you see that rejection is significantly higher when they have these high coefficients of variation for both TAC and sirolimus, that self-reported non-adherence is very high, and most importantly, pursuant to what Peter said, the donor-specific antibody is significantly higher in those patients. Those are going to make a problem for long-term outcome and for subsequent grafts. With that, I have hit my time, and I want to thank you all for your attention, and I'll turn the podium back to the moderator. Thank you very much.
Okay, again, Sarah Eggers, I got tasked with continuing on. They liked so much what we did in the morning that they want the same sort of facilitation in the afternoon. But to everyone here, this is an experiment. But I think we can do this well. Before getting in, I would like the folks, the presenters, the panelists up here who did not present, if you could introduce yourselves so that we know. So.
I'm Marie Chisholm Burns. I'm the Dean and Professor at the University of Tennessee Health Science Center at the College of Pharmacy and Department of Surgery at the medical school.
I'm Bill Fitzsimmons. I'm with Astellas Pharma Global Development.
My name is Kevin Longino, and I'm the CEO of the National Kidney Foundation. I'm also a kidney transplant patient for 12 years.
We asked Kevin to be up here to serve as a voice representing and reflecting the broader patient community. And so you can jump in at any point reflecting that, but we also have you here in the audience to provide the individual context of it. If there are any folks still on the web, feel free to pull in comments as well. So there are 3 discussion panels, and I understand we can go till 3 o'clock with this. If the one person signed up for open public comment, if you can come find me at the break, and we will discuss something. So we get to go for 30 minutes on this, and we have 3 questions to get through. And I think how it's going to be best to try to do this is a little bit of that lightning round format that we did at the end of the topic, the morning discussion, where let's take the first question, how well do we understand the extent of non-adherence in patients post-transplantation? We had 4 wonderful presentations that I have to say, I'm going to speak, I think, for my colleagues as well. They were so well coordinated. Thank you for that. They really flowed together very nicely. We heard a lot from you about the extent of non-adherence and the type. And I think by type, I'm going to interpret this as the whys of non-adherence. Is that a fair interpretation? So, how much is it happening and why is it happening? And I'm going to ask just the panelists, maybe we'll start with those of you who hadn't yet spoken. Let's change it. What, all the things you've heard or what you're in your research, what concerns you the most? And just briefly describe that concern, either probably about the type of adherence or how well we understand the extent of adherence. So for example, it could be the aspect of wanting to live a normal life, or other factors that have really resonated with you of concerning, and then we can get some discussion going on those factors. Okay, did the experiment sound make sense? So let's start with, I'm going to put, Marie, if you could, what surprised you, what concerns you the most when you think of the types of why non-adherence is occurring?
I think what concerns me the most, especially at the beginning when I started researching this, probably about almost 20 years ago now, is that how complicated it could be and how individualized it really is. And I think that that's what we heard this morning from the recipients here, how complicated and how individualized. And with the complication, how much sometimes us in the healthcare profession, it's hard for us to pinpoint or even slow down to recognize it, as well as having the time to do it, and how we need a multidisciplinary team to help with that, and how we actually really, really, really need you guys to tell us about that.
Okay. All right. William, do you have any thoughts? What concerns you? What's resonated?
Rita mentioned the difficulty in defining what's the critical level of adherence that's necessary, and I think that's an important component, because we all say we would like perfect adherence, but none of us are perfect from that perspective. But is taking 80%, 90%, 50% of your medications, and this morning someone mentioned it's not always going to be every 12 hours. Can I go plus or minus an hour around that, 2 hours, 3? We don't have answers to those questions, and those are really important ones.
So both knowing it and then being able to communicate it back to the people living with the organ transplant so that they're not making up their own sets of rules about, well, I think I can go 90%'s okay. Maybe it is. maybe it's not. Kevin, anything?
Well, there were so many good things that were said this morning. It's really hard to add to that. But I think the point that was just made is true. I think a lot of patients try to make their own guesses as to what's tolerable and what's not. And it would be important to know those things. I think in terms of compliance, one of the things I haven't heard us talk about, we talk about medications that have to be taken on the spot at 12 hours. But it's not just that. It's oftentimes you have to have an empty stomach for 2 hours and an empty stomach for 1 hour afterwards. And then if you make a mistake there, then it throws off the rest of your day. And so what happens if you did have a cookie accidentally and it's now inside your 3-hour window? Is that okay? Do you take your medicine at the regular time, or what? And nobody seems to have answers to that. And yet that's a big factor of the compliance and adjusting just the logistics of the lifestyle.
Okay. Thank you. A rich amount of consideration. So let me turn it to the audience and on the web. Feel free to comment. Take one thing that you've heard and say, let me tell you why that's such a challenge, or let me tell you why that's not a challenge for me. So we're not going to be able to take all the comments that we want to, but we'll
really quick thought. Thing I thought about earlier, Nicole Jefferson. Most of us here are here because we're passionate about our organs and we want to keep them. I think a lot of the issues with non-adherence comes from people who don't really think that much about their organs, and those are people who don't have a support system, don't have the family. So I think the families being a part of the doctor's appointments is a very big part, because if a family member doesn't understand that it's important for me to take these medications, I may not remember, and they may not understand why they're important. So I think we have a good grasp on taking on our meds and understanding it, but it's the population out there, especially in my community, that doesn't have the support or doesn't realize the importance of taking the medicine.
Okay. You raise a very good point that I think we all know, which is that you in the room, you are reflective of a patient population, but you're not completely representative of the patient, the entire population of people living with an organ transplant. We know that. So even as you still have challenges, and so we still want to hear those as well, but that is an excellent, excellent point.
I think Dr. Chisholm Burns, you hit it on the head. It's a very complex issue, right? So it's not going to be like one little bullet's going to solve this. And to me, I think if you look at if you're kidney recipients, you have a very poor health literacy for CKD. It's one of the lowest health literacy rates out there. So recognize that from a communication standpoint, because so much just comes down to communication, right? So how much of it, and to be frank, is that the doctors are really struggling right now with the time they have, but it goes back to a message that's appropriate to the audience. And the audiences are not all monolithic. But to me, it goes back to that. It's about the communication, trust, all the basics, and relationship building.
I'll take a couple more.
I would just add that part of the issue is actions are louder than words. And so in the medical profession, for example, you say, take your medications on the 12 hours the best you can. And then you go in for a blood draw, and you're not allowed to take your medications, and you're made to wait until 3 hours beyond the 12 hours. Well, then obviously it's not that critical. Secondly, we're going to do some testing, and you can't take anything, include your medication, for 12 hours before. And then you get there, and next thing you know, it's delayed, surgery is delayed, stuff like that, throughout 18 hours. And obviously, it's not that critical then if we can wait 18 hours. And thirdly, when you're in the hospital, forget any timing on your critical medications. I don't care if it's diabetes or it's immune suppressants. You're on their schedule, not yours. And you can't convince them that this is really important. And by the way, this is my 12 hours. I'm sorry, this is when we give out meds. The action speaks so much louder than the words.
We'll take one more from Piper.
I think that engaging patients in their own care is also a really key part of it. I think that, in my experience as someone who has CF, one of the key components to getting people to take drugs is involving them in understanding how they're going to impact their bodies. Because if the only experience you really have, the only thing you see the drug doing is causing you stiff muscles, for example, then you're going to assume that that med is bad, and I think it's very important to sort of explain, especially when there are multiple immunosuppression therapies on the table, why do I have to take Prograf and mycophenolate and a steroid? Why is it not okay for me to miss any of these? And I think that additional level of empowerment also makes people sort of take control over the medication for themselves, so that, we can stand up to a hospital, for example, that says, well, we're in the ER, so we can't order your meds, and you'll be here for 12 hours at least, and you have to be able to say, oh, well, then I brought my own. And that sort of, you can't do that unless you understand why it's so important.
Thank you, Piper. I think, Rita, you have a question?
I was going to make a comment before. I think that as a pharmacist, I can say this, we're kind of obsessive compulsive and we want, we're trying to give you all these little details of exactly how to do it perfect, when really and truly we need to step back and look at the immunosuppressive regimen and ask the patient, what time of the day does an immunosuppression regimen work for you? How do we build it around you? Yes, all of these things that we tell you to do 2 hours before, 1 hour later, with food, without food, creates the ideal circumstance. But we do, as you said, have ability to adapt that regimen and monitor the drug levels as such that go along with that. However, it requires consistency on the patient's part as well, so we can adapt and adjust to those particular changes. And I think that when we talk about we really don't know the percentage of which patients need to be compliant, I think that just as you do to yourself, we say 100%, knowing that it's never going to be 100%, and and we're hoping that we minimize the lack of the non-adherent times that we can. So, we're learning, but and we continue to learn.
Any other on the panel thinking of question 1 about the whys of non-adherence that it's a burning question for you, or a burning comment you'd like to make about that? Go ahead, Mary Amanda.
I was struck by the comment that Jack had made this morning. I don't know if Jack is still here. Oh, there you are. About how if a medication is missed, there's nothing that you immediately notice. And it can lull a person into thinking, well, if I'm not going to notice anything, it can't be all that important. It's important, sure, but it's not that critical, like if you had some immediate issue. And so I think it gives, sometimes it can give people a false sense of confidence, and without having enough understanding of the real consequences that might not be observable for a while, it becomes a problem. And then the second thing is, I'm concerned about all of the people that are not like you all, that are not here, and how they can be reached, that because they don't see this as a critical issue. You're here because it's so important to you, and you've thought about it a lot, but what about all of those other people that don't think about it a lot? Like, how are they managing? How are they figuring out? Or maybe they're not figuring it out, and they just don't even realize that it's such a big issue.
Peter?
So I think one of the other things that comes into what's been discussed and the point you raised, we don't understand how much non-adherence is acceptable or in terms of tolerated and not have a bad outcome, in part because we're treating everybody as if they're the same. And you made the point earlier, what we really need to get to is personalized immunosuppression, and we honestly don't know how to do that yet. Short of what I showed you with the twin transplants where you don't need any drug, there are some people who don't need as much drug. And I showed you the clinical trial that we did with the NIH, where we withdrew the tacrolimus in a number of patients, and we stopped the trial. But we stopped the trial because some people rejected, but there were other people that came off, and they were fine coming off, right? So that's the other problem here, is that there's a biological difference between everybody who's getting their transplant. Some need more drug than others. The problem is I don't have the magic wand that tells me who does and who doesn't. And that was the whole point of the trial. Could we predict who could come off one of their drugs and try and get to a personalized immunosuppression regime? And we couldn't do it because none of the things that we thought were going to predicted it. So I think that's the other complexity here, is that non-adherence is probably okay in some people. In other words, it gets to some people need less, but we can't predict who that person is. And I think that's some of the challenges that we face and that the drug industry faces, is how do we actually identify and move to personalized immunosuppression? Because I think that is part of the solution here, is allowing patients to realize how much they really need for the transplant that they've received.
Thank you, Peter. And Robert?
What you're all hearing when you're hearing that the patient needs to take the medications at 12 hours on an empty stomach, etc., what you're really hearing, and I see transplant patients sort of every day, what you're hearing is our insecurity as physicians, because we don't know how much is enough. We also don't know what the difference is between patients, and data that Peter has generated suggests that depending on some very sophisticated sorts of matching that we don't do yet well enough, that's coming, that some patients may be more privileged than others. So unfortunately, we do have a one-size-fits-all policy, because that's about the best we can do. So what you're hearing is the insecurity of the physicians, because we think we know that perfect adherence is the best that we can hope for. So that's what we tend to shoot for. The other thing I want to say is that the issue of not having any specific issue when you miss a medicine, an immunosuppressive medicine, becomes a major issue in adolescence, to the point where I was thinking of floating to the FDA that we spike all of, because it doesn't hurt when you make the medications. So I was thinking that we spike all of the tacrolimus with heroin, so that if you miss your medication, you go into a bad withdrawal response. You'd be surprised. Everybody's laughing and says, what a good idea. I don't think the FDA will go for it, but they're here, and so we can see. No, I take that
All
back.
right, so Robert, what you're bringing up, I think, is going to tie into the next question. And before we do that, let's hear one more from Kevin.
Well, just a general comment. What would it take to start a personalized medicine initiative for transplantation?
What would it take to start a personalized medicine
Initiative.
initiative? Yeah. I think it's a broader question. Okay. So let's move on, and then we can always come back if we have time, but let's move on to question 2 about the role of healthcare providers in this. We've talked a lot about healthcare providers today, but this question sort of, I think, wraps it up. Are healthcare providers appropriately involved when it comes to promoting adherence, or are they not paying enough attention? And what improvements would you suggest? And I'm going to start with 2 the panel members, and then we'll open it up. So if we can start with Mary Amanda, do you have any? Okay, wait.
My view,
Okay, let's
start with Peter. Okay.
So it really comes back to, I think there's been a really good discussion this morning about individual autonomy. And what are we trying to create? Are we trying to create a situation where patients are relying upon themselves and developing their own systems and strategies to have autonomy, in essence, versus us trying to assume that autonomy? And I think from one perspective, I would say that we try to be too much assuming your autonomy, and we need to elicit you being autonomous and working a solution that would then be something that works for you. And I think that actually would be a better strategy. So from one perspective, I'd say we're maybe too involved, and that may be leading to problems in and of itself. Although I could argue the other side, and maybe Mary Amanda, I'm sure she will.
They argue both as well, but I guess I tend to think that the problem is not that anybody wants the healthcare provider to hold everybody's hand and hand out their pills all the time and be in their house. It's that they don't ask enough when patients come back. They don't show enough interest often. They assume that things are going okay or that there's no issue if the patient doesn't have any questions. And by not showing interest, I think sometimes patients, and all of us have been patients, feel that then the person doesn't care about that issue. And they're given, I know in our program, they're given logs, they're told they have to record certain things, and then after a while, the program staff just never ask about that anymore. And the person says, why am I doing this? And the staff's view is, well, they're supposed to be doing it for themselves. But what's the point of endless little pieces of numbers written on a page if the provider never looks at that and says anything about if it's okay, if it's not okay, is there something that it's not being understood? So in that sense, I think there needs to be more involvement in the sense of being committed to talking with the person about how things are doing, not holding their hand, talking with them and coming up with new strategies as needed.
Can you hear me now? What if I move over here? So let's ask a question this way to get at thinking about what you just heard from Mary Amanda and Peter. I'm going to ask first, can can someone or a few of you think of the best physician you've had with regard to the helping you as one of your support in your support network, addressing the challenges of adherence? Anyone want to talk? We have Dan over here, and then we'll go to Layla. And what was it? What did they do, and how did you feel about it? Were they really involved? I guess two is not, if you like this, but were they involved? Were they laid back about it?
So I'm not thrilled with this question the way it's worded, to be honest, because healthcare providers is such a general term. Does my dentist really care about my transplant medication adherence? I'm not so sure that he or she is. So the question to me is which one of our healthcare providers are adequately engaged in our medication adherence, and which one of our healthcare providers, maybe who should be, aren't? That's, I think, a more interesting question. But I come from a transplant program that's large in nature, and I really need to take a moment to credit the post-transplant liver coordinator nurse that I have and the nursing team. I can't say enough about what they do in terms of patient engagement, because they are the lifesavers behind the brains and the technicians of the doctors. It is that liver transplant coordinator, Samantha, who is my advocate, who is my person who's checking to make sure that adherence is done to the best of her ability. And because of her, and because of her sort of follow-up skills for me personally, that's what has a huge influence on my adherence. So if you were to sort of take that a step further, you then would have to sort of say, okay, so in order for Samantha to be more effective at her job, is her patient list, and I believe it's hundreds of patients long. So how can someone like Samantha be effective across a large pool of people like that? So what is that right number of patient transplant people that she can engage with? What is that right number to possibly lift adherence to medications and things like that? And then from a suggestion standpoint is I would say that this here, the education I got today in adherence and the things that all of you have brought about is so eye-opening. But I also think we need to find ways to get that to every transplant patient in terms of suggestion to improve our overall education of how important adherence is, because 11 years out, it's still a very important topic to someone who's 11 years out or 11 months out. So those are just a couple quick comments I wanted to make. So thank you very much for that.
Okay. We'll go with Layla.
Layla again. For me, I would say specialty pharmacy at Northwestern Medicine in Chicago. They're on call, basically 24/7. They call me a week in advance to make sure I have all my immunosuppressants, my medicines. And I live in two different places right now, nine months out from transplant, working full time. And there are days where, I have to split up my medication between two different locations, and they've been very instrumental in just being able to coordinate that with me. And then also Robert Edinger, Dr. Robert Edinger, he really supported what I've been feeling as far as, I'm now 28. The last time I was healthy, I was 19. You may see that as an adult, but at 19, I was an adolescent. And I was very non-compliant. I'm not going to lie, like, oh, I was so compliant. No, I was dealing with trying to be normal and not wanting to tell my friends, and I want to hang out, and I'm in college. And then now to be healthy again, and I find that those issues with scheduling and all the things that you deal with as adolescents, because I spent 8 years of my life fighting kidney disease, I haven't fully developed mentally. And now I'm 28 and I'm thrown back into this real world of being healthy. And I'm like, what do I do with this? I don't have these skills. I gained a lot of wisdom and skills on dialysis, but now to, be 28 with basically the mindset of a 19-year-old is something that I'm kind of struggling with. So he really supported that for me. And everything that I've learned here today from everyone helps me to move forward and feel more motivated that I can definitely be far more adherent.
We're going to take one more here, and then I'll have a show of hands questions, and then it's important to get a little bit to the last question. So Pam.
I'm Pam Duquette. I'm Lindsay's mom. And something that struck me was a lot has to do with the patients not understanding that they can self-advocate. A lot of people, and it's been touched on before, where they just, okay, I'll do what you tell me to do, and that's it. I think a lot has to do with the healthcare providers or the centers. At our center, and it's a very large center, every person that we spoke with, and this is over 8 years of her course of her disease, and I'm talking from the some of the pharmacists I actually would speak with, to the nursing staff, to the pre-staff, to the dialysis nurses, everybody always said, are you okay with this? Do you understand this? Do you have any questions about this? What do you feel? There was a lot of back and forth, which when we were both thrown into this, I was petrified, and I just sat there with deer in headlights, didn't know what to do. But it was the constant prodding in a very gentle way, and the fact that they respected my opinion and advocating on her behalf. That was really important.
Okay. So to wrap this up, we'll do a show of hands, and anyone up here can raise their hands, and in the audience. Now I know I think you guys were assigned those cases, but what's the reality for you? Who's making a stronger case, whether it is the provider who wants to give you more autonomy that you're dealing with, or the one where you wish they're too much. They're too much in your face about it, if I can use that term. So, how many think that it's too overbearing? The healthcare providers, and I think we've learned that we need to think of it broadly across the spectrum of providers that you're doing. That basically that you agree with Peter. Okay. What about Mary Amanda? Okay. Too overbearing. Okay. Too overbearing. That the providers collectively are too overbearing about adherence. Okay. Is underbearing a word? Too passive. Not asking you enough questions about adherence. Raise your hand. Okay. Well, it could be that it's just a very hard question. I think what we're hearing is that most people would have sided with Mary Amanda in the description and in terms of what you would like to see. So we'll leave it with Robert.
But in the pediatric setting, the adolescents will tell you to a person it's too overbearing. I had a full head of hair before I started taking care of adolescent transplant recipients. And I can tell you that every fellow, every time, every doctor, every nurse makes attempts to make that connection with the adherence. And the kids get tired of hearing it after a while. So we have to dial exactly how we do it. So again, it's not one size fits all.
Great. Thank you for that point. Renata.
And that's actually a great segue. Let me try to tackle, I guess, Kevin's question and then perhaps try to touch on Nicole's comments earlier in the morning. You talk about individualized medicine, and I guess I'll take the liberty to follow Bob's comment about our insecurities, the clinician's insecurity knowing the doses. The FDA-approved drugs called Prograf, Cellcep, Myfortic, Nulogix, etc. They come in certain strength and certain regimens are recommended. But if you think about it, I'll sort of venture to say that personalized medicine is in its baby steps, because your physician doesn't prescribe you a fixed dose every day, the same dose, and just lets you go. For certain of them, the calcineurin inhibitors, you're getting your levels measured early on after transplant more frequently, later less frequently. Rita talked about medical adherence, which is not a passive act. It's a dialogue between the clinician, be it your primary physician, be it your nurse coordinator, be it whoever is taking care of you. And I guess I've heard also people saying, I ask what's my value. You know, gentleman said, I'd like to know what my what my creatinine is. Can we get a device approved for that? So I think if you think about the drugs, your interactions with the clinicians, asking about your values, and then hearing how your regimen is getting modified. We heard, I don't know if it was James who said, I'm getting belatacept. Doctors are making changes based on the patient's individual course. And I think that, in a sort of simplistic way, is the beginning of personalized medicine. Hopefully in the future, as we learn more, there'll be more of that. But I think the feedback from you in the audience, the patients who are saying, I want to know more, I want to understand, I want to know why. Someone said, I want to know the mechanism of action. Why am I being asked to do this? How does it make a difference to me personally, from my organ, from my lung, from my kidney? And then the FDA approves the drugs that I mentioned, but economics came up. So as you are aware, they're not present today, otherwise I'd ask them to speak, but there's a whole group called the Office of Generic Drug Products, and they're responsible for making available to the public generic versions of the products that start as innovators. The goal of that group is to make those products, I'll use the phrase carbon copies. They should have the same effect, the same efficacy and safety profile as the innovator, because they really are trying to be carbon copies in terms of their therapeutic activity. Anecdotally, we will hear that patients don't do as well. Can I ask for people who have those experiences, what Sarah said earlier, please share those experiences. Perhaps we can look into that. Every so often, a drug company will have a problem in the plant. Something goes wrong. The tablet is not full strength. That's a solvable issue. But that's different from, maybe a patient didn't do well because they didn't take it the same way, because it didn't taste the same way, or they couldn't swallow it the same way. It's unclear. But the only way we will know is if you find out something or you hear something, you send it to the FDA. And again, let me make the plea that Sarah voiced earlier. Until November 27th, everyone here, everyone on the web, if you have information that you think we would find valuable, like we have all the discussion that we've had, please send it in.
Thank you, Renata.
And that may be a segue to your
Yeah,
last.
at the break, we will put the information on how to get to the docket, which is your way of submitting a comment electronically. So I think we want to make sure we address the last question quickly, and then we will go into a break. So we'll do it at kind of a lightning round style. First of all, can I have show of hands? How many of you have participated in a clinical trial? Okay, we've had some experience here. Okay, so unfortunately, I don't think we get into what might make it difficult or hard about measuring adherence in the clinical trial. You can write that in the docket. But let's get anyone on the panel who wants to address this question briefly about how critical it is to collect the adherence data. I think we heard from the panels, but any other thoughts on that or perspectives?
So maybe just from the concept that if you're trying to qualify a new drug and you end up showing it doesn't have a benefit, but if the patients weren't taking that drug or adhering to the regime of that new drug, then you've essentially disqualified a drug, but it might actually have had an effect had it been taken appropriately. And that's the risk of a clinical trial, not knowing their adherence activity around that drug. So ideally, you would know for both arms of the trial what the adherence rates are, and that then you're actually judging the drug on its effects as opposed to the non-adherence, which may show that it has a negative effect.
Okay. Any other thoughts? Yes, William.
And the other side of that is if you're performing a study that is a non-inferiority study, rather than failing to show the effect of the drug, you may make two drugs look very similar because they're muddling the two arms of the trial. So there are consequences either way depending on the design. But I think it's important to keep in mind the clinical trial setting, particularly in the registration-driven trials, is very different than the real-world setting. We're already out there having patients sign informed consents, putting them on a specific schedule, counting the medications when they bring them back and when they're dispensed. It's a very different setting, so we have to be careful in realizing that that's not immediately able to extrapolate that to the on-market real-world setting outside of the trial.
Okay. Can I ask a show of hands of the number of you that have participated in a clinical trial? Was adherence, the challenges with adherence, easier when you were in the clinical trial? Harder when you're in the clinical trial? Anyone, we can take probably 2 comments on anyone's experience having to be adherent while on a clinical trial and challenges that you faced? Okay, we're putting you on the spot, so I think we will take that is a sign that, I'm going to take it as a sign that it's a break time. So we will take a break and come back at 3:15 and continue with